Hey everyone, and welcome to the video talking you through some of the past paper questions on proteins. I should add that I'm now uploading worksheets on my TES shop, and the link for the shop can be found in the description below this video. I thought it would be useful to share those sheets so that you can download them for free and work on them before watching videos like this to help you see the answers and approach for the questions. Okay, so let's get going. Question 1a asks you to describe how a peptide bond is formed between two amino acids, and this question is worth 2 marks. This means we have to make 2 points as part of our answer. We know that the reaction that forms a peptide bond is a condensation reaction, so the first marking point will be awarded for stating that it's a condensation reaction. You can also draw a diagram to show this reaction if it makes your life easier. The next marking point would be to state where the condensation reaction takes place, so we need to state the functional groups of NH2 and the carboxyl group. You could also say that the reaction takes place between the amino group and the carboxylic group for this second mark. Part B expects you to describe how the secondary structure of a polypeptide is produced. First of all, you need to think about the bonds present. We know hydrogen bonds are what holds the shape together, so that would give you 1 mark. And the next mark will be awarded if you successfully state that the hydrogen bonds are between the NH of one amino acid and the CO of the other amino acid. I actually think that it's easiest to state that the hydrogen bonds occur forming the beta-pleated sheet or the alpha helix. For that second marking point, you might want to mention the beta-pleated sheet or the alpha helix to make your life a little bit easier. I certainly would recommend that you say this over where the hydrogen bonds are. Part C of this question was for 2 marks, and they asked you to explain why two proteins might have the same number and types of amino acids but have different tertiary structures. If you think back to that analogy I used with the beaded chain or the necklace, each of those beads could be in a different order as you're putting that chain together. So the first marking point would be related to talking about the primary sequence. In that sequence of amino acids would be different. You could even say that the primary structure is different. We also know that if the sequence is different, then the ionic hydrogen and disulfide bonds that form it in this particular tertiary structure will form in different places. That then gives you the different tertiary structure. So hopefully that answers that particular question. Question 2 gave you an image of the amino acid serine, and it asks you to identify the R group on the image by drawing a box around it for 1 mark. The second part asks you to draw circles around each of the parts which would be removed when 2 other amino acids join directly to it. If we look at the image, we know that the R group will either be at the top or the bottom of the middle carbon. We also know that the middle carbon is always bonded to a hydrogen, so we therefore know that the R group must be opposite the hydrogen that's in the middle carbon. With the second part of the question, it says 2 amino acids would join, so we need to put circles around the H and the OH of this particular serine molecule. The red box and the circle show you the correct answers for these 2 marks. The next part of the question asks which 2 substances are formed when 2 amino acids are joined. We know that water is one of them, and you should also state that the dipeptide is the second substance formed. And finally, for 1 more mark, they ask you to name the type of bond formed between the joined pair of amino acid molecules, and you already know that this is a peptide bond. So far, so good. Nice and easy. I'm not actually going to go through all of the questions, but I have picked 2C for this video as it's really similar to 1C. In this question, they ask how the change in the primary structure may result in a different 3D structure. We know that if the primary structure changes, it is the sequence of amino acid that's changing. We also know that means that the protein will fold in a different way due to the bonds forming in different places. By bonds, we mean the hydrogen, disulfide, and ionic bonds. Some students sometimes say peptide bonds form in different places, and this would be rejected as the peptide bond positions do not change. Question 3C is the next one that I want to go through. This time they give you a diagram of glycine and ask you for 2 marks to draw the dipeptide produced when you join 2 of the glycine molecules together. This is basically asking you to draw the condensation reaction. The first thing I would have done is to draw the glycine a bit like this. Note how I've drawn the NH2 differently from the diagram given on the exam paper, and same with the COOH structure. I've basically just split it up. I find it easier to draw it like this so I can see all of the hydrogens separately. I've drawn two glycines because, of course, we're drawing a condensation reaction between both glycines. The next thing I would do is draw a box around the hydrogen and the OH that are going to react together. And then I would draw the peptide bond between the carbon and the nitrogen. This is the structure of the dipeptide overall. I have changed the position of the hydrogens to be above the nitrogen and the carbons just for the neatness. I would also label this bond as the peptide bond, and I would add the H2O at the end to show that I understand that it's a condensation reaction and I understand the position of the bonding. Feel free to pause the video now to copy this structure out as it is on the screen in case you didn't get this question correct. The next question is an extended response one, where for 5 marks you are asked to describe the structure of proteins. I would start right at the beginning and talk about how proteins are made from amino acids, and these amino acid monomers are joined by a peptide bond formed by condensation reactions. I would then describe the primary structure being the sequence of amino acids and how the folding forms a secondary structure in terms of the hydrogen bonding. You could, at this point, also talk about the alpha helix or the pleated sheet formation if you don't want to talk about the hydrogen bonding. They would accept that as an answer. If I'm still continuing on this question, I would then talk about the tertiary structure and the 3D folding due to the hydrogen bonding as well as the ionic and the disulfide bonds. And the final marking point could be awarded if you talk about the quaternary structure being more than 2 polypeptide chains. Now, please note that for this, it is only 5 marks, so you don't need to mention all of these points. The reason that the mark scheme gives you all of these points is a good revision aid. I guess what it helps you do is to plan out your answer and have a thought pattern as to the different things that you could talk about. The next question is question number 6. You have a passage that they provide to you. The first part of the passage talks about coming into contact with poisonous substances and how the skin acts as a barrier. The next paragraph talks about the skin being the largest organ of the body and how the outer layer consists of keratins, and that keratins are a group of proteins that differ from each other in their primary structure. It goes on to say that each keratin molecule consists of several polypeptide chains, each individual chain wound into a spiral or a helix. And it also talks about sulphur-containing amino acids. What you have to do with questions like this is use the information from the passage to answer the questions. Lots of information in passages like this can be unnecessary to you answering the questions. So the skill comes from sifting out the important information in order to help you answer the questions. The first part of the question actually asks you to evidence from the passage about how keratin molecules have a quaternary structure. So I've left the answer on the screen here. We know that quaternary structure proteins are proteins that have more than one polypeptide chain in the molecule. So that's what you needed to state for this 1 mark of this question. The next part of the question asks you to explain how sulfur-containing amino acids help to give keratin molecules their characteristic strength. Well, we know that where there is sulfur in amino acids, we know that that allows the formation of disulfide bridges. So stating that will give you 1 mark. You could even say for the 1 mark that the R groups form stronger disulfide bonds. The second part of the mark is awarded for saying that the bonds, the disulfide bridges, will bind the chains to each other. Most students miss out on this second mark as they don't always talk about the chains binding to each other. Part C asks you to explain the differences in primary structure which results in keratins with different properties. Well, this is really similar to some of the other questions that we've seen earlier in the video. The key here is to either say how the primary structure could be different, so it could have a different number of amino acids, or it could have a different sequence of amino acids. Either of these would give you 1 mark. The second mark is to state that the bonds are in different places, which gives you a different shape. The final question gives you a bit of information about the degree of hydrolysis and an equation. Part A says That the protein molecule contains 151 amino acids. What is the total number of peptide bonds present in this molecule? Well, we know that when we have two amino acids, for example, there is one peptide bond between them. So by having 151 amino acids, we must have 150 bonds, i.e., one less than the number of amino acids present. Part B gives you a DH value of 18 and it asks you to calculate the number of peptide bonds that have been hydrolyzed. We know that the total number of peptide bonds present is 150, and we know that from part A of the question. So we simply need to rearrange the formula. 18 multiplied by 150 gives you 2,700, and then you divide by 100 to get 27 bonds that have been hydrolyzed in total. The next question asks you what the DH value would be if it were completely hydrolyzed into amino acids. This would mean that all of the bonds would need to be hydrolyzed. We know we've got 150 bonds, and if we plug that into the formula, we would effectively be doing 100 times 150, which is 150,000, and then dividing again by 150. So overall, you're left with the answer of 100. The explanation for this is to say that the 150 bonds and the total number of bonds present cancel each other out in the equation, and therefore you are left with 100 as the DH value. So hopefully that makes a bit more sense. Mathematical questions make up about 10% of the overall paper when you sit your AS or your A2 papers. So it really is important to become comfortable with the mathematical content of A-level biology. We will talk about this in other videos as we go through. So I hope that was really helpful to talk through, everyone. Remember that if you're looking for the worksheet questions on this topic and the other topics that I've already covered, you can find them for free in my test shop, the TES shop, where you can download them and answer the questions. The link for the test shop is in the description of this video, but you can access that shop by simply searching for TES in capital letters, Bioteach London in Google, and the first result will take you to my shop. These videos are aimed at helping you understand what the correct answer is, as well as help you in your approach to these questions. So by downloading these worksheets and trying them out yourself first, and then watching these videos, hopefully you will be able to grasp the approach a little bit better. Thanks for watching this video, and don't forget to like the videos and subscribe to my channel. See you all very, very soon. Bye for now.